Dear viewers, today we will learn the practical on histology of pituitary gland, ovary, testis, adrenal gland, uterus, seminal vesicles. Aim of the practical is to study histology of pituitary gland, ovary, testis, adrenal gland, uterus, seminal vesicles. Introduction. Histology is a branch of microscopic anatomy and deals only with the microscopic structure of tissues. Different tissues combine to form organs and organ systems. So, for the structural and functional details of the organ and organ system, it is necessary to study histology. Histology is thus important to study normal structure of tissues and its comparison to pathologic conditions. First, we will see histology of pituitary gland. The pituitary gland, also known as the hypophysis, is a bean-shaped, roundish organ that lies beneath the hypothalamus, resting in the base of the skull called the sella turcica. It is composed of two distinct parts. First is the anterior pituitary, also known as adenohypophysis, is a classical gland composed predominantly of cells that secrete protein hormones. Second is the posterior pituitary or neurohypophysis, is not a separate organ but an extension of the hypothalamus. It is composed largely of the axons of hypothalamic neurons which extend downward as a large bundle behind the anterior pituitary. It also forms the so-called pituitary stalk, which appears to suspend the anterior gland from the hypothalamus. These two areas have different embryologic origins. The anterior lobe, that is adenohypophysis, develops from the ectoderm of the pharyngeal roof, that is oral cavity, that is to pocket called Rattke's pouch. On the other side, posterior lobe, that is neurohypophysis, is derived from the floor of the diencephalon. Both parts of the pituitary show different structure. The anterior pituitary shows a highly cellular structure. The neurohypophysis, that is posterior pituitary, has a fibrous structure. Now, we will see histology of adenohypophysis. First part is pars distalis. In microscopic level, the pars distalis of the anterior lobe of the pituitary contains bundles and cords of epithelial glandular cells which are polygonal in shape. Among these clumps, walls of highly vascularized connective tissue are present. Using hematoxylin eosin stain, these cells can be differentiated into two main groups. First is chromophil, those cells which are colored with this stain technique. Chromophil cells can be subdivided into two groups. First is acidophil. These cells are characterized by a dense pink stained cytoplasm. Second is basophil. The cytoplasm of these types of cells is stained in light purple. Second type is chromophobes. These are those cells which don't stain in this technique. Acidophils and basophils are divided into different classes of cells which have different secretory products and target organs. The subtypes of acidophils and basophils are described in the table below. Second part is pars intermedia. In this layer, colloid filled vesicles are present. Beta cells, other secretory cells and chromophobe cells are present. Some cells of the pars intermedia produce the melanocyte stimulating hormone, that is MSH, which causes increased pigmentation of the skin. Endorphin cells produce adrenocorticotropic hormone, that is ACTH. Neurohypophysis. In microscopic level, the pars nervosa of the neurohypophysis shows a very fibrous structure with some sprays cells among the fibers. Using hematoxylin eosin stain, these cells are showed as irregularly shaped cells with oval shaped nuclei, that is called the pituicytes. They are astroglia related cells that can be stained with glial fibrillar acid protein, also called as GFAP. Among the nerve fibers can be seen oval shaped poor stained structures called herring bodies, which stores hormone filled vesicles. Neurohypophysis releases two octapeptide called oxytocin and vasopressin, that is antidiuretic hormone. Ovary. The ovaries are the female gonads responsible for the formation of ova. They also produce hormones that are responsible for the development of the female secondary sex characters and produce marked cyclical changes in the uterine endometrium. Each ovary is an oval structure about 3 cm long in diameter. Ovary is lined by a cuboidal germinal epithelium. Beneath the surface epithelium is a layer of dense connective tissue called tunica albuginea. The innermost layer of the granulose cells surrounding the oocyte forms the corona radiata. These cells are more columnar than granulose cells. Between the corona radiata and surrounding oocyte is a non-cellular glycoprotein layer, the zona pellucida. Stromal cells surrounding the follicular cells differentiate into theca interna. At the stage of follicular development at the theca externa, the cell layer outside the theca interna has not differentiated. Numerous primordial follicles are located immediately below the tunica albuginea. Each primordial follicles consists of a primary oocyte surrounded by a single layer of squamous follicular cells. The developing oocyte has a large eccentric nucleus with a conspicuous nucleus. In growing follicles, the follicular cells proliferate by mitosis from a layer of cuboidal cells called granulosa cells and surround the primary oocyte. In large follicles, the follicular cells change to cuboidal or low columnar. The large follicle before ovulation is called Graafian follicle. Testis. The right and left testis produce the male gametes or spermatozoa. Each testis is an oval structure about 4 cm long. Each testis is enclosed in a thick connective tissue capsule called the tunica albuginea. The tunica albuginea consists of closely packed collagen fibers. Internal to this capsule is a vesicular layer of loose connective tissue called tunica vesiculosa. This connective tissue surrounds, binds and supports the seminiferous tubules. In the posterior part of testis, tunica albuginea expands into a thick mass called mediastinum testis. Thin fibrous septa present extending from the mediastinum testis toward the tunica albuginea, dividing the testis into numerous compartments called lobules. Each lobule contains 1 to 4 seminiferous tubules. These are long and highly convoluted. They are arranged longitudinal or tangential planes. Numerous blood vessels are located in the interstitial connective tissue around the seminiferous tubules. Clusters of epithelial interstitial cells, also called as Leydig cells, are present in between the tubules for secretion of the male sex hormone, that is testosterone, into the blood stream. Seminiferous tubules are lined with specialized stratified epithelium called the germinal epithelium. They consist two types of cells, the spermatogonial cells that give rise to the spermatozoa and Sertoli cells. Sertoli cells rest on the basement membrane of the seminiferous tubules and give nourishment to the spermatogenic cells, in addition to support other proteins and hormones. Adrenal gland. The right and left adrenal gland lies in the abdomen close to the upper poles of the corresponding kidneys. The adrenal gland consists of an outer cortex and inner medulla. It is surrounded by a thick connective tissue capsule in which are found branches of the main adrenal arteries, veins, nerves and lymphatics. Sinusoidal capillaries are found throughout the cortex and medulla. Adrenal medulla. Both functionally and embryologically, the medulla of adrenal gland is distinct from the cortex. When the gland is fixed in a solution containing salt of chromium, as an example potassium dichromate, the cells of the medulla show yellow granules in their cytoplasm. This is called the chromaffin reactions, and the cells that give a positive reaction are called chromaffin cells. It is made up of group or columns of cells which are separated by wide sinusoids. These cells are columnar or polyhedral and have a basophilic cytoplasm. Medulla also contains sympathetic ganglion cells arranged in small groups or singly. They exhibit the characteristic of vesicular prominent nucleus and a small amount of peripheral chromatin. Adrenal cortex. The adrenal cortex is made up of cells arranged in cords. Sinusoids intervene between the cords. On the basis of arrangement of its cells, the adrenal cortex is subdivided into 3 concentric zones. First zone is zona glomerulosa. It is outermost layer of cortex. It constitutes the outer 1/5 of the cortex. In this zone, cells are arranged as inverted U-shaped formations or acinus-like groups. Small columnar or polyhedral cells are present with basophilic cytoplasm and deeply staining nuclei. Second zone is Zona Fasciculata. It forms middle 3/5 of the cortex. The cells are arranged in straight columns which is 2 cells thick. Sinusoids intervene between the columns. Cells are large, polyhedral with basophilic cytoplasm and vesicular nuclei. These cells are very rich in lipid that can be seen by lipid droplets, which gives vacuolated appearance. Third zone is Zona reticularis. It is the innermost layer of cortex. It is so called because it is made up of cords that branch and anastomose with each other to form a kind of reticulum. The cells present contains darkly staining lipofuscin pigments. Capillaries in this layer exhibit irregular arrangement. Adrenal cortex secretes glucocorticoids, mineralocorticoids and some amount of androgens and estrogens. The medulla contains chromaffin cells that secretes epinephrine and norepinephrine responsible for fight and flight response. Uterus. It is muscular female reproductive organ. It consists of upper expanded part that is fundus and lower narrow part that is cervix. It is commonly referred to as the womb. It is a pear-shaped organ about the size of a clenched fist. It consists of blood-enriched tissue that slows off each month during menstrual cycle. The powerful muscles of the uterus expand to accommodate a growing fetus and push it through the birth canal. Histology of uterus. The uterine wall is composed of the three layers. First is outer layer, that is serosa or perimetrium. Second is middle layer, that is muscularis or myometrium. Third is inner layer, that is mucosa or endometrium. First layer is perimetrium. It is outer surface of uterus made up of single layer of mesothelial cells. It is supported by thin layer of connective tissue. Second is myometrium. It is a thick smooth muscle layer of the uterus. It consists three layers. First is inner longitudinal layer, second is middle circular layer and third is outer longitudinal layer. Circular layer is very vascular, also called as stratum vasculaire. Third is endometrium. It undergoes cyclic changes that result in menstruation. It consists of simple columnar epithelium, that is ciliated and secretory cells. An underlying thick connective tissue is present called as stroma. Mucosa is invaginated to form many simple tubular glands which extend through the entire thickness of the stroma. Only this layer takes part in the menstrual cycle. Blood supply is from two types of arteries. First is coiled arteries and second is straight arteries. There are several layers of endometrium. First is stratum basalis. It is retained at menstruation. It functions as a regenerative zone for the functionalis after its rejection. Second is stratum functionalis. It is luminal part of the endometrium. It is sloughed off during every menstruation. It is the site of cyclic changes in the endometrium. Seminal vesicle. It is an androgen dependent tissue. The seminal vesicle is a sac-like mass that is really a convoluted tube. The seminal vesicles develops from the vas deferens. Their histological organization resembles to some extent that of the vas deferens. They are elongated sacs about 4 cm long and 2 cm wide which taper where they unite in the vas deferens. Each seminal vesicle consists of one coiling tube about 15 cm long. All the lumina visible in section of the seminal vesicle are in continuity in the intact organ. It has an outer covering of connective tissue, a thin intermediate layer made up of smooth muscle and an inner mucosal lining. The mucosa shows thin, branched, anastomosing folds. Goblet cells are present in the epithelium. The structure of the epithelium is variable, appearing columnar or pseudostratified columnar, that is columnar and basal cells. The lamina propria of the mucosa is fairly thin and loose. Distension of the seminal vesicle by its secretion shortens the mucosal folds and the columnar epithelium. The ducts of the seminal vesicle transport the strongly eosinophilic secretion to the colliculus seminalis. The secretory product of the columnar cells, which may be seen in the lumen of the seminal vesicles, is strongly acidophilic. They secrete fructose, globulins, ascorbic acid, prostaglandins, citrate, inositol, cholinesterase, a number of soluble proteins, inorganic phosphorus and acid soluble phosphorus. It contributes 70 to 90% of the seminal plasma. These compounds, which is released by the seminal vesicles in addition to fructose, has three main functions. First is the formation of the sperm coagulum. Second is the regulation of sperm motility. Third is the suppression of immune function in the female genital tract. Summary. At the end, to conclude, in this topic we have studied histology of various tissues like pituitary gland, ovary, testis, adrenal gland, uterus, seminal vesicles. Their structural aspects are important to correlate with their function. Further, study of their structure is necessary in pathological status of the tissue. Thank you.